Okay, so hi everyone and welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping housed in the Martinos Center. It's my great pleasure to introduce our speaker today, Dr. James Duncan. Dr. Duncan received his bachelor degree with honors from Lafayette College and his MS and Ph.D. both in electrical engineering from UCLA. Dr. Duncan has been a professor of diagnostic radiology and electrical engineering at the Yale University since 1983 and has been a professor of biomedical engineering at Yale University since 2003 and the Ebenezer K. Hunt Professor of Biomedical Engineering at Yale since 2007. He has served as the acting chair and is currently the director of undergraduate studies for biomedical engineering. He has published over 300 peer-reviewed articles. He is the Life Fellow of the IEEE and a Fellow of the American Institute for Medical and Biological Engineering, as well as the MICAI Society. He has served as co-editor-in-chief of Medical Image Analysis, as an associate editor of IEEE Transactions on Medical Imaging, and on the editorial board of multiple other journals. He is a past president of the MICAI Society, and in 2017 he received the Enduring Impact Award from MICAI Society. I would just like to remind the audience to please address any question you have using the Q&A box. Professor Duncan, thank you so much for coming here today. The virtual stage is all yours.
Great. Thanks very much, Or, and thanks for the invitation. And it's great to be with you all, if not virtually, if not in person, at least virtually from above my garage down here in Madison, Connecticut, where it's raining, but hopefully I heard it's nicer up there, says Or. So I'd like to talk to you today about our work in our group on neuroimage analysis and autism. And this is a collaborative effort between our image processing and analysis group, mainly Larry Stabe, Nisha Dvornik, Zhentang Zhuang, and Xiaoxiao Li, who's now moved on to be a postdoc at Princeton, and myself. And working with Pam Ventola, who's a child psychologist and faculty member in our Child Study Center who studies autism spectrum disorders. So, as I'm sure many of you on this call know, I'm going to be talking about autism and the spectrum of disorders. But I will start with a sort of trying to define it roughly for our purposes. Then talk about some initial image analysis work early on with some structural work, but initial ideas in looking at functional differences between ASD and typically developing controls using a basic fMRI task-based analysis. We moved on from this to look at group data, as I'll mention, and trying to look also at subnetworks and grouping by communities. And this was a traditional, I guess it's called these days, Bayesian estimation strategy, but I think you'll see how we built the work. Then moving on to our latest work, which is, as I'm sure many of you are doing, data-driven learning, for both initially for biomarker detection, so classification, and then discovering biomarkers from that using first convolutional neural nets, and then our latest ideas on using graph convolutional nets. And then finally, for about the last third, which is really our goals with a lot of this work, is to think and look at prediction strategies to try to look at treatment outcomes, especially to certain cognitive treatments like this pivotal response treatment, and try to see how, predict how people or subjects might do from pre-treatment or baseline fMRI. And then I'll try to draw some conclusions and wrap up a bit. So the autism spectrum of disorders is really characterized by three sorts of impairments and difficulties: social interaction deficits, repetitive behaviors, and language impairment. But there's a variety of comorbidities, especially hyperactivity disorder, anxiety and mood, or certain even depression disorders. And there's a variety of other sorts of things too that may come into play. So it's a spectrum, a wide range of symptoms and severity. It's quite prevalent in the US. I think this number is up to about 1 in 45 children become afflicted with ASD. And it is more common in boys than girls. Researchers are trying to understand this better, but about 4 times so, at least as of today. And the cause overall is unknown, so therapists and psychologists tend to treat symptoms. So our earliest work, our initial ideas, actually, we used just that we were a segmentation-oriented group, as many of you know, and sort of looking at structural segmentation and measurements. So not shown on this slide, but we looked at things like trying to segment the hippocampus or the amygdala and look at volumetric changes. But our key collaborator back in the early 2000s was Bob Schultz, who's now a known investigator in autism down at University of Pennsylvania. And Bob brought us into the fMRI world for these studies and this work, and started initially looking at this faces houses task. So for task-based fMRI on looking at autism subjects versus control subjects, basically young male adults. And this was on our old 1.5 Tesla magnet in the MR Center that Todd Constable and Doug Rothman run. And so these first tasks were faces, houses in 24-second alternating increments. And what Bob found, and we helped a bit with this work, was if you looked at the activation regions based on looking at GLM residuals and t-tests, you ended up with, in the typically developing young male adult group, that both the parahippocampal place area and the fusiform face area, as shown here, lit up. But however, in the autism group and the ASD group of young male adults, basically the fusiform area was not, the face area was not lighting up, implying that perhaps the subjects were not responding to this biological form of a human face, that they saw faces and houses just as objects or places, if you will. So this inspired further thinking and further work. And really, we were both what was going on and still going on today. There were hypotheses and interests in thinking about looking at subnetworks in the brain and how they might change due to autism. Our main collaborator in the Child Study Center now is Kevin Pelfrey, who we still work with, even though he's also moved on to University of Virginia. But as we started this work in the early 2010s, we were fortunate to have Archana Venkataraman come to our group as a postdoc. And she had done her Ph.D. with Paulina Golland over at MIT, and is now a young or a faculty member at Johns Hopkins. And Archana looked at this problem a little bit differently. I really enjoyed working with her, and I was sort of thinking about it a bit differently. So we still had task-based data, but she said, let's think of the whole problem as if we now look at connectivity matrices and sort of think of those data as noisy observations of some underlying generative process that we would try to find by estimating parameters in a latent template that was some hidden organization. So these probabilistic interactions we could sort of pose and try to understand the information, all with task-based fMRI primarily, and also we were generally looking at group data. So here was the strategy that Archana posed. And we basically, the data were these connectivity matrices done with Pearson correlations. And we used the 150 regions based on the Destrio atlas. And I was just looking this up, and as an aside, we use different atlases in the work I'm presenting today. I guess it's almost like a greatest hits of the Bruce Fischel, Anders Dale Atlas catalogs, but you'll see it move around a little bit, but we started with this one, the Destrio Atlas. And so Archana, we looked at data from both normal controls and did correlations of the 150 regions of themselves and called those data B, capital B, and B bar was in the autism spectrum group. And so with the region labels pre-assigned by the neuroanatomical Destrio Atlas, the idea was then to estimate these sorts of templates, if you will, of synchrony, or basically connectedness. So each node was ROI in the atlas, and then connectedness was its correlation or connectivity to all other nodes in that template. That would be connectivity F, and F bar was for the ASD group. But the idea was that we'd sort of discretize what Archana called synchrony or connectedness into just several discrete, even binary values of low, medium, and high, or ultimately low and high synchrony. So we're trying to estimate this latent variable. And furthermore, could we estimate what community? So we pose just a few communities, actually 2 or 3, sort of relating to hypothesized subnetworks with a few non-random prior parameters. And we try to estimate an abnormal community type here, YK. So the problem set up is a joint probability density function where you have the probability of the region type in one abnormal community or not, or one of the two, or just normal, the synchrony parameters and the connectedness parameters, given some declaration of how many abnormal communities there were and something about their parameters. So this is all set up and solved as a maxima posteriori estimation problem and used expectation maximization to then estimate the synchrony parameters F or F bar and whether R is normal or belongs to one of the abnormal, just several abnormal communities. In this work, we moved the tasks to from faces, houses to these, what they our collaborators in the Child Study Center call biopoint forms, basically a human form on the one hand, or scrambled motion patterns of these light dots on the other hand, and alternating again 24-second increments. And so we had in this work 72 autism spectrum disorder children and 43 age-matched controls. And we posed or hypothesized 2 communities. Remember, these are group data, and you're looking at different views of the brain here. And in the pink community, sort of imposed on these sort of puffed out, blown up brains, are the first a hyperactive or a hypersynchronous community. And this is where the connectedness or the synchrony in ASD was greater than that in the normal subject group in a particular region. And a blue hypoactive community or hyposynchronous, where there was reduced connectivity in ASD, F bar less than F. Now, we don't have ground truth really on this, but we did, as others in the field we were observing had done, was we took the neurosynth database with all these regions from all these papers. It was nicely figured out about different function and brain activity, and took these regions and just mapped it onto that to try to get some sense if what we were doing was plausible. And interestingly, the hyperactive community, remember more connectedness in ASD, mapped onto strengths, or we thought perceived strengths of pattern recognition and maybe visual perception, but less so in anything to do with social interaction or emotion. Whereas the hypoactive community, where there were strengths and normals, this matched more onto social emotional processing. So encouraged by this, we started off onto our next set of work. But remembering that this was all done, all this analysis is just group analyzed data. And what we really wanted to do was to look at individual subjects and map those onto things like neurosynth, or just in general try to interpret things. And so we started off in this direction and wanted to do individual subject analysis. So our initial work on this was with Xiao Li, my grad student, who I said is a postdoc at Princeton and actually is about to start a faculty position out at British Columbia. And this was sort of a classification, set up as simple classification of either ASD or control using convolutional neural nets, again from task-based fMRI data, and then to try to look at the decisions and do underlying biomarker detection in the regions. And, you know, we were pulled towards data analysis or data-driven ideas for a variety of reasons, because there's a lot of interest and excitement in the field. And also I would say that I don't think any graduate students would work with me if they weren't doing something on deep learning or neural nets. So, I mean, I'm kidding, but it's almost somewhat true. So again, here's our biological task on the data sets we used here. Now we're, and I forgot to mention in Arch and his work, we were also using the 3T Siemens magnets in our MR center. And here's the same biological motion task in the scrambled motion, the person's sideways now, but the same idea. We had, because there was no treatment in this initial group, 82 ASD children and 48 typically developing controls. Pre-processing was done in very similar ways that all of you have sort of almost pioneered and helped develop. I guess, motion correction, interleave slice timing correction, brain extraction, spatial smoothing, and high pass filtering. And we made sure that there was no head motion effect between the groups. And we had about 146 time data sets in 3D. So here's Xiao Xiao's work. It's basically, if you look to the left of this dotted line, it's the basic architecture. So we had 3D, 146 3D fMRI time series volumes, and we ran them through a convolutional neural net classifier and ended up then with a distribution per time point and per test subject of a probability density approximation, I guess, density function into classification to ASD or control. Now, what we wanted to do, though, is try to figure out which regions were most important. So we did this with a corruption analysis. So we took region by region and corrupted it, adding noise, and ran it through the same classifier, and then compared it to the results of the original classifier, looking to see what degraded the performance of the classifier, and then declaring that as an important region, as you'll see. So we did that region by region and then got this saliency map out. So here's the basic setup initially. So we started with 146 time points, which were 146 3D 2-channel image volumes, because we took a 3D, a 3-frame sliding window, and for each voxel basically computed the mean and the standard deviation of the residual fMRI time series in that voxel. So we took what got from 146 volumes to 144 volumes, average over 3 time slices, and each channel at each voxel then was a mean and a standard deviation. And this earliest work was back in ISB 2018. So the architecture, those of you working on data-driven learning architectures, you'll recognize a pretty standard architecture. But at that time when we started it, we thought it was fairly unique, and we were interested in getting into convolutional nets. So we took these 2-channel inputs over the 144 time points and ran them through a 6 convolutional layer net with 2 fully connected layers at the output. Had dropout of a half after layers 1 and 2, and a little more after layers 3 and 4. And then got 256 fully connected nodes at the output, and then did a sigmoid thresholding, and ended up with the probability of ASD or normal, basically probability of ASD having 144 values, real values, 1 for each time point. Somewhat, simply, we just thresholded it a half and ended up with 144 binary values of 1 for autism, 0 for control, and just used the majority vote wins sort of to try to help with ideas like overfitting. And so we compared the classification alone to different ways of doing regressions with logistic regressions, support vector machines, random forests, and felt pretty good that the F scores were getting up there for this test. And so we had 130 data sets, as I said, distributed amongst ASD and normal. Each fold did a 5-fold cross-validation on this, where each fold was 26 subjects. Now, the corruption saliency mapping, trying to find which regions were most important, was a little more involved. As I said, we used the Destry-O atlas for this work, and we parcellated into those 150 regions, and then corrupted the original values by resampling and replacing regional values from the training data, and did this corruption ROI by ROI to try to determine which ROIs were most important. And basically, if the prediction distribution makes classification into ASD or control more difficult, then we declare that region as being important. And this initial work was in MICI 2018. So that's what got us these distributions. Each distribution, again, in a test fold was 26 subjects, 144 time windows, so about 3,700 or so values in these graphs. And the idea was then we watched what happened to the distributions of ASD versus TDC when a different region is corrupted. So and so basically it grouped into these 3 situations. if we got these nice class distributions, if we corrupted a region and the ROI caused the two distributions to move towards each other, then we felt that that region was contained features useful for identifying both ASD and control. If the ROI had patterns associated mainly with ASD, it moved the ASD distribution towards the normal, and vice versa, that the control moving towards the ASD made that most important. Now, this is a bit, as you can tell, a little bit obviously qualitative here. So we tried to quantify it and do it reliably looking at a Jensen-Shannon divergence criterion and then categorizing direction of shift by a Wilcoxon sign rank method and decide whether it's one of these B, C, or D. So here's the average over all 26 subjects in a test group, a single test group with 16 ASD, 10 TDC. The colors sort of map out onto these different parts of the brains shown here. And we did map these onto the NeuroSynth database, and the region classifications as decided. And so blue, again, reminding you, is equally salient ASD and control. And to some extent, at least, it mapped onto these sorts of regions in default mode kind of regions. The red regions that were felt to be more salient for ASD classification, somewhat more onto the language and visual strengths. And then green regions related to controls, and less for the ASD, more related to emotion and social processing. So we were encouraged, and this was, remember, on individualized data, so we could do it subject by subject. But one of the things you might notice here is that there is no like implicit ideas here of trying to cluster into communities like the earlier work on the group data that we did with Archana's ideas on the community detection. So we wanted to get that incorporated here, and wanted to move forward with these ideas in general. So we had a variety of thoughts to extend this work. We did try to get away from the cluster or the distribution movement ideas and try to use cooperative game theory as an alternative to region corruption, using something known as Shapley value explanation from that. But that was kind of complicated, I would say. And I guess Xiao who did this work, Xiao Li, was feeling that the field was starting to look more and more at graph-based methods. So we moved in this direction for a variety of reasons, as you'll see. And it, got away from the region corruption process. And we felt we could take advantage of the natural underlying graph structures of the ROI maps from the different kinds of neuroanatomical atlases. And there was a bit more straightforward interpretation to the co-function of the multiple ROIs that we might be able to build in. So this moved us towards thinking about the brain ROIs as graphs. And so this next section then is again from task-based fMRI for individualized biomarker detection, only now using graph convolutional networks. These are the papers from, just in summary, from this earlier CNN work that I just talked about, convolutional neural nets. And here's the initial publications on the graph neural nets approach in MICI 2019 and 20. We're in the midst of revising a paper for medical image analysis that summarizes this. So here's the thinking here. So again, back to the greatest hits of the Fishville-Dale atlases, we moved to the Desicon-Kiliani atlas, which only had 84 ROIs. A lot of reasons for this. We were looking to better manage the number of ROIs, I guess, for the initial work was probably the key motivation. And from this atlas, each ROI basically is declared as a node in a graph, so a node V. Then on each region, we computed 100 features on each. We'll call those node features H. And this did include the bold signal mean and then ROI, the bold signal standard deviation carrying over from the earlier work, and then did a full Pearson correlation to all the ROIs. It gained a lot more features, and as well as the ROI coordinate. But the edges, we needed a strategy for this, and we went to partial correlations, a more true correlation. And so sort of correlations built in twice to this, I guess you could say. where the partial sparser correlations, perhaps the true correlation, because the correlations between regions are controlled for all other regions, were used to determine the edges in the graph. And then we did have to do augmentation by resampling. Our numbers were still not all that large. So as you'll see, we did that too. So here's the full processing chain for what we call brain graph neural net, brain GNN. We have the fMRI time series, just like before. We have brain parcellation, only now using just the 84 ROIs from the Desikan-Kiliani Atlas. Mean, or there's time series data in each one of those regions. This functional correlation matrix really is meant to show correlation overall, but it's basically both the Pearson used in the node features as well as the partial correlations used for the edges. And we get this graph, and then we want to run it through our brain GNN processing that I'll talk about in a second, and kind of pull all that together and make a classification decision, while then underlying, inferring what the most important information is in the communities, and pull out biomarkers. Now we could use this directly for prediction, which I'm not going to show here. We talk mainly about the classification biomarker detection. So you might say, why graph neural nets? And I think we would say that sort of for 3 or so reasons. One is scalability. We can process an arbitrary number of ROIs or regions as we feel, so we can move to more regions if we wanted to. It is pretty effective in that we can exploit the natural data structure of a neuroanatomical atlas for the graphs. And it's efficient in that the computation and processing is distributed and maps nicely onto GPUs and distributed processing. And also, to some extent, the graph convolutional net and graph neural net can be more directly interpretable. So here's our brain GNN computation. And so we take the basic graph net and we run it through 2 graph convolutional blocks. And then there's a readout layer here, and then other processing to do the classification to summarize. Each one of these graph convolutional blocks is made up 1 green and 1 yellow block. One to do basically, we'll call it processing on the nodes and on the features rather, to reduce those. And another one is sort of a pooling layer that then reduces the number of nodes. So to be clear then, the node convolution layer, the node features change, but the graph structure doesn't change. And on the pooling layer, the node features do not change, but the graph structure changes. So as we go through the different blocks, we start out with maybe 100 features and 84 nodes, then down to 10 features and 42 nodes, and cut the nodes in half and down to 5 features to do the final computation and classification. So first I'll talk about this layer that we call the region-aware convolutional layer, or ROI-aware convolutional layer. And we would just point out that in a traditional graph convolutional net, basically all the nodes are treated identically, and you more or less apply during learning the same weight kernel or same kernel to all nodes. But when we have nodes that have spatial relationships, like we do in our work here, we wanted to impose this ROI-aware module. And this is where we're going to try to reinsert the community assignment ideas. And we could just start off with a random community assignment, but then learn a conditional kernel embedding where you have membership into different communities. And the communities are going to be basically characterized by a stated assumption of how many communities we'll have in the beginning. And during training, we'll learn the communities and their membership, and basically declare that beta sub u actually is a community basis function, and alpha will be the membership of any ROI I in community U. So the embedding really does a rectified linear unit extraction of a set of weights, Wi or Wj in the raw form, that are weights on the features themselves for any node, and a weight on the edges relating neighboring nodes. But as I say up here, instead of learning just WI and WJ directly, we impose this notion of mapping into a space where there are these community basis functions that are learned beta, and then alpha membership of any region onto that community within an offset B. So beta I, beta 1 through beta K, with a stated or declared number of communities are what are learned. And instead of learning the WIWJ, we learn alpha, beta, and B, which relate to community assignment membership into these different basis functions or basis vectors, if you will, that are also learned from the training data. So here's our initial shot at it on data. We're back to using 72 autism spectrum subjects and 43 healthy controls. And we used, really, it's just a declared number of communities of about 8. Our sister lab with Todd Constables worked on the different ideas here, and including on the human connectome and other things. And so we just use an approximate number that's in the literature. And declare 8 communities as being interesting or relevant. And then the idea is to then take the 84 ROIs and get them set up through the learning into, or we set up the communities through training and learning, and then on the forward thing on a test subject, try to declare which by alpha values, what the membership is of any test community and ROI into one of the 8 declared communities. And here it is on one of the subjects, BioPoint ASD versus healthy control, and looking at the underlying regions grouped into communities. And so we don't have the neurosynth analysis on this yet, but we feel we're seeing plausible communities as we do this, and plausible membership of the ROIs in these communities as we do this work on different test groups within this overall training and test set. So the second idea on the second module that was on the right back here, sorry, let me go back, this pooling module now is also a key part of all this. And so we basically, we want to now reduce the number of nodes if we can. And we use this, so after the first module, in the first stage, we're now getting down to perhaps 10 features at a node. And we want to do a pooling action to sort of project the vector feature information onto a normalized pooling vector, a virtual pooling vector, in order to do the pooling and the comparison, and ultimately end up with node scores from which we can just keep the top K scores. And then reduce the number of nodes. And so we do this top K pooling, and it's nice because there's no regulation on pooling scores, and the ranks may be different for different input instances, but we can control it with this regularizing and normalizing idea. So the overall GNN loss function, then, I'm putting together all the modules overall, is a cross entropy loss for the class, basically autism or normal, and then 2 other terms, a distance term and a so-called group level consistency loss that are built into the loss function for training. So classification loss built into that is the encouragement of the community assignment into one of the underlying community basis functions. The distance loss tries to, it maps onto this pooling and tries to push away through the top K loss less important regions and keep important regions in order to trim down to this fewer regions. And the group level consistency loss, that's taken from the training data, and it really regulates the similarity of different subjects' pooling scores. So it's sort of a cheat in a sense. It reincorporates something about group level or training level group consistency, even though we're still doing individualized estimation of classification and regions. So here's 3 subjects out of our test group of 72 ASD and 43 age-matched controls. These are all ASD subjects, looking at them from the left and right side. This is subject 1, subject 2, subject 3. Again, here's the loss function, and this is just the consistency with the group analysis in that region. And basically, we're just looking here at the lambda 2 term, how if you keep that small or zero, you see that these are what the important regions that come out from the analysis and are important for classification. And this is more individualized. As you crank up lambda 2, these regions and their kind of interrelationships become a little smoother. So that might be a good thing. So lambda 2 can be used as a more common ROI group biomarker. So that's, I guess the classification results are shown here then. And so we had 72 ASD subjects, 43 controls. We augmented the data by resampling and replacement 30 times. Used 3 folds for training, 1 for validation, 1 for testing. So we had 690 graphs times 5 folds, 3450 graphs. And here's just the classification results, not commenting further on the regions. And so we compared it to support vectors, random forests, a BrainNet CNN alone that was out in the literature, our own CNN without graphs, an interesting graph idea called Graph Sage. I think it's out of Stanford, our own initial GraphNet and this new net. And so we got pretty good classification. These are the number of parameters in thousands that need to be learned. So it's lower here. And you know, as you get up to a full CNN, there's a million parameters. We're somewhere in between with about 41,000 parameters that need to be learned, but we're encouraged by this. So this is all then so far about classification and biomarker detection. And that's where we sort of are at with this work and this graph-based work for classification, biomarker detection. So the last part of the talk, about the last 15 minutes or so, 10 or 15 minutes, I'd like to talk about our work towards prediction. And here now we're trying to predict response to different kinds of mostly cognitive therapies that are applied to children with autism. And this is Pam, our collaborator, and her daughter who does not have autism, just showing the idea of this pivotal response therapy that is really intense therapy done over 4 months, 5 or 6 hours a day with the parents and the therapist. And so, or the therapist. And so no one size fits all. It's meant to bring out early our social skills and interactions. And the idea is, from baseline fMRI, can we help do some prediction of what might work and what might not with this treatment post hoc? And this pivotal response treatment is the key one that Pam and our group's been looking at. It was developed in the early 70s. So again, the baseline imaging then is done with the same biopoint human form and scrambled motion forms, again in the 3T magnet. And these are the acquisition parameters, about 3.5 millimeter cube voxels, the biopoint scramble. Can't get this to turn off. In 24-second increments. These are very golden data, and we only have so many of them. Only 21 children as of 3 or 4 months ago were brought through this treatment because it's so involved in the 16 weeks. But we did have 14 males, 7 females. And we had both pre-treatment and post-treatment severity measured with this ADOS score, the Autism Diagnostic Observation Scale. These scores, and most importantly, the one we looked at is the Social Responsiveness Scale, or SRS score. And so the idea was, could we predict treatment response, basically change in SRS between post and pre-treatment, based on the pre-treatment task-based fMRI scan, with the idea that lower SRS, by the way, means better social function. And the hope is then that the predictive modeling could help guide the treatment assignment, or at least be useful in encouraging parents to go through the treatment. So our initial work was using random forest, and we did just sort of a standard thing here of taking fitting general linear models, using the residuals and getting a t-test between bio and scrambled motion. So we got t-statistic images as the input to the initial work here. And Nisha Dvornik, my colleague, former postdoc, now colleague, has worked on this using random forest. And so the idea was, can we predict treatment from pre-treatment imaging using t-statistic inputs and random forest? And this early work was pretty brute force in a sense. We just decided that this template of images in the sort of the frontal cortex and a few other regions, the amygdala and ventral striatum, would be the regions we would choose. We selected out about 270 voxels to get started. And the idea was then to train random forest trees and then predict change in SRS score from any given subject. So here's the sequence of this. We started off with 267 voxel variables. We used out of bag error then, so that after you train them, take the non-trained samples and run them through the trees and try to see what was most important. So when the higher the score, when leaving things out, the more important the variable. So we got this down using the t-statistics to about 13 top-ranked variables by looking at out-of-bag errors. Used this again to do some linear bias correction, and started off with about 1,000 trees in this random forest training, again on these fixed regions. And we only had 19 subjects that we started with here. And really, at the end of the day, in this work, there were only about 13 voxels that were used, so pretty kind of crude, if you will. But we did get the predicted residualized change in SRS versus the actual change. We got a pretty good correlation, although brittle, right? Not that robust, and everything was done pretty manually by, at least initially, to extract the regions. So Juntang Zhuang sort of built on this, and we tried to say, okay, with same 19 or so subjects, can we have a high dimensionality? Can we go through a two-step process where and then we started with a atlas that Xiwen Shen and Todd's group, Constable's group, was developing that had 268 regions, parcellate based on that, but select just a smaller set of regions and extract some voxels within those regions. So using random forest again, but a two-stage process. So we had 268 region predictors, 19 subjects, or really it was used to leave one out. So I think 18 to train and one to test on, and had 268 variables, but introduced 50 or so noise or shadow variables on top of that. Started with 2,000 trees and just generated these noise things as independent random noise. And then at the end of the day, calculated the importance of each region using, again, out of bag error, and basically discarding variables that were less important than the noise variables. So this trimmed it down to about 500 trees after region selection. So got it down to 500 trees to begin with, and then used just voxels within those regions using a gradient boosted method. So really ended up with about 10 important regions and hundreds of voxels. And here is the map using this method. We're down automatically selected, but down about 0.71 on the correlation of the predicted change in SRS to the measured. These were the regions that were selected as important, where blue was positive, red was positively related to the treatment outcome, and blue negatively. But this was, and sort of similar regions we found were important, again, using the out-of-bag error to judge the importance. And so this is sort of the importance map to get that. But all of this was not that robust, to be honest. This was a little bit better. The random forests were a bit tedious, I would say. So Nisha Dvornik and our group, we sort of said, let's move towards thinking about more advanced sort of and more data-driven neural net architectures. And so we selected to go, and here's a different atlas again, sorry, the automated anatomical atlas, but to keep the number of regions down about 90. And now we use this idea was now basically built on recurrent neural nets based on, because we wanted to preserve a kind of order of temporal information. So this used these long short-term memory modules, and we now incorporated somewhat crudely, but interestingly, both task-based fMRI and some phenotypic data as input into the learning algorithm. And the goal is again to predict or regress out change in SRS score, and then use that to look at the correlation to treatment outcome. So the learning algorithm, the recurrent net now, was made up of a bunch of these LSTMs, as you'll see, one for each different time point. And for those of you not familiar with it, maybe many of you are, basically these LSTM modules are sort of modules with a bunch of trainable weights organized in this sort of input-output pattern. And there's really 4 sets of layers that need to be learned: a forget layer, an input gate layer, an update of the current state, and an output layer. So we use these at different time points, as you'll see, and how to learn those parameters. Now, the number of data sets were low. We did use 90 ROIs as input to each time point, but we did a sampling and replacement again to bootstrap and augment to get the data up. In fact, we did it 50 times and just sort of randomly selected time series within different regions and replaced the information. And we did this data augmentation about 50 times. So here's the final architecture. Again, now reduced to, well, about 156 time points with 2-second increments between them. Remember, it's the human biopoint scramble or light pattern versus scrambled light. So we did that task, both of them about 6 times over to get 156 time points. Because it was task-based, the individual subject data was basically lined up. And at each time point, with just a little bit of averaging, you had 90 input values from the 90 ROIs from the AAL Atlas into each one of these cells. Each LSTM cell then was made up, or each one of these cells basically had 8 LSTM nodes, and each node had the 4 sets of weights that I talked about earlier that need to be learned or trained on. So, and then furthermore, we tried different ideas, but we felt that the most straightforward was to take the simple phenotypic information and use it to initialize the first cell in this sequence. And because this was time-ordered data, we felt we could kind of move through the sequence, go through a fully connected node, and use that to regress out the change in SRS score, percent change. So basically the phenotypic variables, which are only 4 of them that we use, could initialize the initial hidden state in the dense layer. And then these initial states are passed to the first cell that then, with everything propagated through the network. So here's the results, the earliest results on this. If we just use the fMRI data and try to run it through the net and do prediction of the true SRS change in this social responsiveness score, we got significant correlation, it was only 0.35. If we add the augmented data, we got it better, up to 0.53. But very interestingly now, initializing by the phenotypic variables, and now it's age, sex, full IQ, and baseline SRS, it did do a pretty good job of getting our R values significantly better also than augmentation to about 0.77. So to be clear, we had just 21 subjects, but we did augment the training subjects by 50 and got about 1,000 training subjects. Again, there's 90 regions into each LSTM cell with 156 overall. So I guess I'd suffice to say we're just encouraged by this, and it's somewhat early in our prediction work. In fact, where we're going, and this is what we're proposing to do next, is we want to pull together the spatiotemporal information and the ideas. And if you look at just the right side here, this is the same prediction module, but instead of putting in raw ROI-based time series data, we'd like to run windows through our brain GNN graph neural net thing to pull out important features and important nodes and use that as an input into each cell at different window time points. So it becomes this hopefully integrated, we call it dynamic spatiotemporal graph, and then try to get an output here in terms of prediction. We can use the same basic architecture to do classification and to try to look and pull out the importance scores of the biomarkers and use those as attention into the next module, or use them as identification of important regions in general. And I mean, there are harkens to lots of other work in the literature, probably, including work at MGH, but other work by, for instance, Vince Calhoun in his chroniectome ideas of trying to look at spatiotemporal information. So that's it. Just want to summarize by saying that we tried to show both traditional and deep learning, meaning both convolutional net and graph convolutional net methods for biomarker detection, As early efficacy ASD markers, it's basically anchored around a task-based fMRI with the BioPoint data. We did look at a variety of machine learning strategies to try to predict response to this one particular behavioral therapy, although we're now looking at other therapies for both that include cognitive therapies plus oxytocin, as well as other therapies to look at anxiety. But that our base ideas, including our recurrent nets, are used to inform and move towards personalized treatment prediction. Our trends are that we're using data-driven networks. We're probably there in general, and we're going to stick with it. We want to move towards complete time series. We want to reincorporate, and we did for the biomarker detection, but want to get this into the prediction algorithms. And then we're looking for, can we do transfer learning with resting state data that's more ubiquitous for the task-based work? We want to do more on explainability. Working with Doug Rothman's group and starting to look at GABA and glutamate as possible signals in gross regions with MR spectroscopy data to add to these biomarkers. And the goal is personalized individual subject analysis. I want to thank NIH and INDS for the support, and thank you all for your attention, and more than happy to take some questions. All the best.
Thank you very much, Professor Duncan, for this great talk. As we'll now move to the Q&A phase. And while okay, so I see we have our first question. Just a sec. Okay, so Bruce, feel free to unmute.
Hey, Jim.
Hey, Bruce, how you doing?
That's great. That was fantastic. Love the talk, although I'll have to freely admit that I'm going to take a little while to catch up on the details of the methods, but a lot to think about, and big picture, very, very cool, and I got it. Two quick questions. One, and you already kind of led the way with your comment at the very end about working with Doug Rothman, adding, MRS signals. Are there particular challenges to adding, increasingly high dimensional other data? I'm imagining, say, PET data or something like that, into the mix where, unlike the phenotypic data, which, fortunately is relatively low dimensional, you can imagine PET imaging, now, you've got your problem kind of squared relative to the MR data, or at least to some degree. Is that a particular challenge, or conceptually kind of clearly fits into the framework you've laid out?
Right. Well, the first wise guy answer is we had MRS fully integrated, and it was right before we were resubmitting the R01 renewal. And, we couldn't get people in the MR center because of the whole COVID mess. So we actually couldn't get data, We have a little bit of data. But the more scientific answer, of course, is that I think that MRS in particular, I'm excited about. They've seen some action, I think, in the back of the brain, or maybe you know of this or other work, looking at particular changes in the GABA signal due to kids with reading disorders. And we felt that that was interesting. And we started to look at kids with autism, get a little bit of action. These are much bigger regions, though, right? They're fatter voxels. But that's not a problem, because we could just overlay it on some of the neuroanatomical atlas regions and just incorporate it that way. For initializing these networks too, just like we did with the phenotype information, we could, we looked at different ways of doing this. Should we do it at the input, the output? Should we, augment things? And we sort of ended up with this initialization idea into the networks. And for that, we can do anything you said, sort of. It's just, it might not have the fidelity region by region. So if we're going to add the localization effect, that we've got to be a little more careful about. PET, we have not. So we have a PET center, and Rich Carson, I know him well. We're working on it. We're not doing, radio tracer imaging with these kids, that's for sure at the moment. But I think there are some interesting possibilities. And, because that's now more about metabolism, it really is complementary to all of this stuff. So I excited about the possibilities. The easiest path for me at the moment seems to be the MRS ideas.
Certainly for this population, you wouldn't necessarily rush. Although, in adult, ASD subjects, patients. We've been doing PET, very successfully with the MR,
okay.
and so it's not inconceivable that these same, concepts could
Yeah.
play out, and that was helpful. Just
Yeah.
a very quick other question from the prediction standpoint. This may not be a fair question to ask you, but do you have a sense for kind of how good it would have to be to actually kind of be actionable from a practical standpoint? Have any of your clinical colleagues given you a sense for that?
Yeah, that's a really good question. I would say that even what we've got in the 0.8 range at least can be advisory for parents. That's the main initial thought here, because these, they're, I mean, I don't know, you folks, I'm sorry, I forget if you've done some work in autism, but it's an interesting area because the parents are so welcoming of anything that gives them a hint. And if they're going to go into something that's pretty involved for 4 months, if there's just something that says, okay, well, we got an 80% likelihood that something is going to happen, that's useful.
Motivating.
In full clinical work and comparing different therapies, which is just what we're starting to do now, I think we need it to be better to say, okay, now you want to include oxytocin, and that's because it, gets up another 15% of likelihood of an effect. That's a tougher decision. I don't know what the number is, but probably more than 80% if it's your child and you're going to start giving drugs.
Yeah.
So I'm just guessing that it needs to be in the 90% range.
Oh, very, very helpful. Again, fantastic. And thank you very much for your willingness to do this.
Yeah, no problem. I'm glad to be here.
Great.
Thank you, Bruce. And while other people are typing, I'll just would like to ask like a general question. So we've used like top-notch algorithms, CNNs and LSTM, and basically the graph neural nets. Do you think after all the developments we've seen in, let's say, the recent decade, are we about to reach the plateau of the basic neural net algorithms, or is there a lot of room for developments?
Yeah, well, tough question. I mean, we might, as you know, we might have, I don't know if we reached a plateau. I mean, the main things we've not reached a plateau in is dealing with, explainability and interpretability issues. Even if we get a good classification result, why the heck did it happen? And can we really localize and get back into hypothesize something that's fully explainable to the neuroscientists? So I don't think we're there yet at all with that. I think overfitting, as we all know, it remains a problem. And so, that means, do we just keep getting more data, or how can we do it? I'd like to think too is that some more intelligent modeling and hypothesis driven work can augment these and help control some of the, almost serve as an intelligent regularizer to help with overfitting. So I don't think we're there yet. Are we getting there where we can apply neural nets to everything under the sun and get some kind of a result? Yeah, maybe we're getting saturated on that. But true usefulness, I don't know. I don't think we're there yet. I see one question up. Should I take it from Jacob Hooker?
Yeah, Jacob, do you want to try and unmute?
Sure. That was fascinating to watch. Like Bruce mentioned it, in the deep understanding of what you've done, I'm not there, but I did want to know, with these kind of approaches, to what extent you can actually leverage the heterogeneity of a population, be it autism or otherwise, to try and build in a better appreciation for, segmentation of population diversity. I really struggle with like flattening autism spectrum disorder or many other disorders to a unified classification clinically, and then using that as an inclusion criteria, of course, biases us to this assumption that there's some underlying related causality to the features that we see. So I'm just trying to get my head around, like, as these algorithms and approaches become more sophisticated, what should we be doing to pay attention to that heterogeneity?
Yeah, great question. And in fact, we got inadvertently sort of dinged on a grant review on something similar. So I'm very appreciative of the question. I guess our first thought is on this work was despite what you're saying, we needed to crawl before we could walk a bit. And so we just took all comer, anything that was declared ASD and see what we could do. But now we did try to take baby steps towards at least trying to understand something in the heterogeneity related to comorbidities. So we looked at anxiety as a comorbidity to start with. Dennis Sudolowski in our group is interested in that. So we started to tease out those. And I guess the plan is to then, you know, what we would do would be to train on different groups and see how they, you know, they do or don't work across groups. And now we're starting to incorporate hyperactivity disorders and a bit on depression and mood changes. So, I mean, these are just different comorbidities. Can we at least see what's going on with, you know, can we predict different parameters that are not just something related to, let's say, social interaction, but other measures of anxiety? So I guess now it becomes a multi-dimensional problem of here's ASD or ASD with different comorbidities. Here's different outcome parameters. Can we generate a matrix and try to understand what, you know, if we train with more subparcellated groups, what does that mean in terms of the outcome prediction? I know this isn't fully answering your question because you're asking about how do we go in from the beginning, but I guess if we're trying to do it on the back end a little bit. Any comment?
No, I think that's helpful in how you're thinking about it. I don't know that there is an answer right now.
Yeah.
That's sort of the problem.
Well, it's funny because I've been working, as you saw, a bit from the working with colleagues studying autism for many years. And I'm sure if you're into this, you know, over these 25 or so years, it started off as one thing, then it became a spectrum, then it became regrouped. So the field itself continues to sort of categorize and redefine, I've learned. So it is dangerous for us as data, image analysis data scientists to drink too much Kool-Aid at any one time point, I guess. But we want to work in partnership with the neuroscientists and the behavioral psychologists to try to figure it out.
Great. I don't see any other question for now, so I think we can conclude with that. Thank you so much again, Professor Duncan, on behalf of everyone, and we hope to see you in person hopefully soon.
Thank you so much, and it's great to be with you all, and wish you all the best. And good to see you.
Thank you again.
Yeah, sure, absolutely. Take care, folks.
Bye-bye.
Bye-bye.
Bye. Thank you.